Does synesthesia have a genetic basis? First, we need to understand what synesthesia is. Synesthesia is a phenomenon where the stimulation of one sense induces a secondary perception in another. It often involves color word associations, like Wednesday is green. Other common examples include numbers and letters being associated with colors. Synesthesia is shown to be more frequent in people with autism spectrum disorder and is currently hypothesized to result from differences in gene expression during brain development.
As found in a recent study performed by a number of collaborating institutions, such as the Autism Research Center at the University of Cambridge and the Donders Institute for Brain, Cognition, and Behavior at Radboud University, these differences are the expression of 6 different candidate genes. Based on the findings of prior studies, this recent study hypothesized that synesthesia-causing variants were unlikely to be shared across different families. To test this, they looked for rare heterozygous mutations that perfectly segregated with synesthesia within 3 separate families that all showed at least 3 generations with sound color or auditory visual synesthesia. These families were identified from the Cambridge Synesthesia Research Group database and were confirmed to have sound color synesthesia using the established test of genuineness.
Exome sequencing yielded a total of 37 variants across 37 genes. No variants were shared across all synesthetes in all families. Exome sequencing is when only the protein coding genes, the exomes, are sequenced. So what are these 37 genes? 6 of these genes have been found to be expressed during early neural development when synesthetic associations are formed.
These 6 genes span the 3 families. Each of these 6 genes of interest has detectable expression in auditory, visual, and parietal cortexes during development. These genes were also all associated with axogenesis, which is the formation of new axons that allows for the conduction of new nerve impulses in the brain. This figure shows gene expression in human parietal, primary auditory, and primary visual cortices from 12 weeks post-conception to the age of 40 years. You can see that these 6 candidate genes are continuously being expressed along this time period.
Overall, the study concluded that there are genetic variants that impact neural development which lead to synesthesia. The study also shows that alterations that lead to hyperconnectivity likely develop during childhood. Thus, genetic variants increasing the likelihood of synesthesia may act in concert with early life experiences to form synesthete's unique secondary mapping.
So why are these findings significant? Not only is this one of the first studies done on the genetic basis for synesthesia using a family-based study, it may help with understanding the genetic basis of autism, because this study supports emerging data that synesthesia shares some phenotypic and mechanistic features with autism. With further research, these 6 genes for synesthesia may provide support for hyperconnectivity between brain regions as an underlying mechanism for causing autism.